Good morning, students.
Good morning, ma'am.
Okay, just give me a minute. I have to quite understand how to navigate this because I'm not able to see the time. So students, before I start the class, can you help me a little bit today? So 10 minutes before the class, can you just let me know that we have 10 minutes left so that I also wrap up the class on time? Because I'm not able to see the time window when I'm on MS Teams somehow.
Yes, ma'am, we will let you know.
Are you able to see
Sure,
the presentation that I have shared? Is it visible to you?
Yes, ma'am.
yes, ma'am. First slide.
First slide. All right. Thank you. So, wait. So, without any further delay, let's start the class. I hope the second slide is now visible to you.
Yes, ma'am.
All right. So, what we have learned so far in the few lecture classes that we've seen that there are some motors called polymerization motors, and we started with a simple example of actin, right? Where actin, and we saw how actin polymerization can actually exert a force and can move a bead. So if you remember the experiment where there was a bead that was sort of held in position in an optical trap, and you had actin polymerization that had begun from the surface of the bead, and then you had a stiff boundary at the end, and so the actin polymerization would proceed and should have ideally stopped at the boundary, right, where it hits the boundary, but it does not. What we found was that the actin polymerization would continue because, first of all, there could be small thermal fluctuations in the filament, and that could give it some space for addition of a second subunit or an additional subunit. and then that would create a force against the wall. But because the wall, the boundary was rigid, what was happening was that this bead was experiencing a force and was moving backwards. And you could calculate the distance moved by this bead, and you could have an idea about the force that was being generated by actin polymerization on this bead. And it turned out to be of the orders of a few piconewtons. Now, if you consider a similar situation in microtubules, when we say a polymerization motor, we not just mean that it is polymerizing, but also it has to be exerting some kind of a force. So here is microtubules once again for you. And as we had studied the last time, it is made up of this alpha, beta tubulin dimers, right? So the repeat unit here is the dimer. And just as a quick recap, you had both alpha and beta that were bound to GTP. And so somebody in the class the last time had raised an issue about GTP concentrations in cell. So who can give me what might be the GTP concentration inside a cell and ATP concentration inside a cell?
Ma'am, I tried to find out using that bio numbers website of Harvard, but the thing is that ATP concentration and GTP concentration are available there in specific cells like E. coli. It is different for E. coli. Erythrocytes it is different, and oval cells etc. It is different, and also it is therefore different organisms.
Okay. Give me
also,
micromolar, nanomolar, millimolar, molar, what range?
Yeah, it is. Units are also different. I have to see. I just a second. I'm telling.
Ma'am, from what
Ma'am,
I
it is
most
millimolar, ma'am.
Okay, thank you. So here it is. You have an excess amount of ATP or GTP always inside the cell, not meaning that you have too much of it, but it is always within a certain range. It does not, as I've mentioned previously, the concentration of ATP or GTP inside a cell never exceeds. It's always within a narrow threshold. You would never see it going too high or too low. Too low means the cell is actually going to die. There's going to be apoptosis or some form of cell death. Too high is also not seen because there is a sort of, because these are all catalytic machineries, right, which produce these molecules. So they also have a threshold. They also have a threshold because there is a certain number of these enzymes. There is a threshold because there's a certain kinetics that they have to follow, etc., etc. So you have a threshold for this. Now, having said that, you have always an excess of ATP or a GTP. We have already seen for actin why actin cannot be always active in a cell. What about microtubules? So microtubules, the concentration of microtubules, if you just see alpha beta tubulin dimers, would be of the orders of some micromolars. Let's say some 1 micromolar, 5 micromolar, something like that. You have millimole concentrations of GTP. If there is something like this, if you have really an excess of GTP, then you might think that there is always a tendency for this reaction to go into the polymerization state. But we know that there are a lot of other factors when it comes to microtubules that inhibits it. What are the things that we have known so far? We know that microtubules, although both alpha and beta bind to GTP, GTP hydrolysis does not promote polymerization per se. GTP hydrolysis at the beta is known to destabilize this lattice structure of microtubules, right? That's what we saw last time. Unlike actin, where ATP hydrolysis is actually promoting polymerization, you have actin in a state where it is very stable inside the polymer. Here, that's not the case. Here, if you have GTP hydrolysis, that point makes the lattice prone to undergo catastrophe. The only way it avoids catastrophe is because of the subunits at this end, the growing plus end, which are still attached to GTP. So if you can sever this, say you use very high power laser induced severing of actin, of tubulin, so that you do not have this GTP gap, you would see an instantaneous catastrophe. The microtubule protofilaments would sort of fold back like this, and there would be dissociation of the microtubule filaments. What we also know is that unlike actin, microtubule nucleation is a highly unfavorable event. So if you have microtubules, alpha beta dimers in solution, they do not like to spontaneously undergo nucleation. So even if you have excess of GTP, it doesn't quite work the same way. Instead, in cells, you have certain structures that allow nucleation events to become favorable and go in this way, which is what we see in microtubule organizing centers. And I had also asked you to look up at the name of the structures that you see in a dividing cell, say in a cell which is in metaphase, where you see that there are beautiful spindles which attach to these chromosomes, and then the spindles move apart, right? They shrink, and you could see the individual chromatids moving away, but the spindles seem to originate from two poles in the cells. So I also asked you to look up the name of these two poles. So who can give me the name of these two poles?
Ma'am, centrosome.
Okay.
Centroids, ma'am.
Okay. Okay. So now
Am I audible,
we have this called the centrosomes, which are made where you have these centriole kind of organizations where you would see short microtubule kind of structures, which are actually at 90 degrees to each other, and from where you can see radiation of these microtubules. And these microtubules, actually, the way they are formed is very beautiful. You will see microtubules jetting out of that structure, trying to, trying to sense their environment, try to catch a chromosome. And the way it catches a chromosome is it goes and it binds to the centromeres, and then at the kinetochore structures, and then you see the receding of the microtubules and the pulling apart of the two chromosomes. So there is a lot of force being exerted there. We'll look at how that force is generated too. All right. And we have also understood that microtubules are unique because they undergo something called, they're very dynamic, they undergo something called catastrophe. And I've already explained that a bit before, where I said that if you have, as long as you have this GTP cap, the structure is kept intact, but the moment the GTP cap is lost, this is how the protofilaments seem to dissociate. They seem to buckle. And this buckling induces the catastrophe or induces the depolymerization of the microtubules. And then the depolymerization will proceed until you have a certain number of these dimers, which you would have it above a certain concentration. And then that would again promote polymerization because you would still have some structures here left. But if before that you have the entire tubulin structure depolymerizing, then the polymerization has to proceed again via either finding out some microtubule organizing centers, which could be like the gamma tubulin ring complex we had talked about the last time, or it could be some pre-existing small microtubule structures. But again, de novo synthesis of or de novo nucleation of microtubules is a highly event. Okay, and here is just a recap of the one experiment we had discussed. So the idea that GTP hydrolysis is not driving the polymerization, not driving the polymerization event, but actually helping in dissociation, comes from utilization of a slowly hydrolyzable analog of GTP that is put here. Now, if you have something that behaves exactly like GTP, but hydrolyzes very slowly, then you should have an extended GTP-like cap, a very extensive GTP-like cap, right? Because it will hydrolyze slowly. And if it hydrolyzes slowly and you have an extensive GTP bound lattice here, and if hydrolysis is indeed required for catastrophe, you will see the catastrophe even being delayed because there was delay of hydrolysis, there is delay in formation of GDP, and if GDP is indeed required for catastrophe, there is also a delay in catastrophe. So that is what they had seen when they used this slow hydrolyzable. It is not a non-hydrolyzable analog of GDP, but a slowly hydrolyzing analog of GDP. Okay. So this structure we had seen previously. We had seen how you could have 13 protofilaments, 15, 16 protofilaments coming together to form the microtubule cylinder. And we had seen that there can be quite a lot of variability in this, but inside cells, usually this 13 structure is preferred. Now, this is another thing. As I had mentioned, microtubules, although we know a lot about microtubules, it's still a field that is being researched. For example, it's not quite well understood how exactly these protofilaments come together to form the microtubule structure. So here there are some cryo-electron images of growing microtubules. And so from these images, it's like taking snapshots of, say, if an alien visits the Earth today and they would like to understand how humans live, then if they take a number of images, then they would see that at any given point of time, human beings are eating, are walking, are talking, are doing a lot of activities. So they can combine those images to understand how human beings behave, right? So it's the same thing. You take a lot of images of these microtubules and you combine them to try and understand how these microtubules might behave. And so there are now a couple of theories, and none of them can be actually disproved or kept away. Maybe it's a combination of all that is happening. So the theories here are that you have the formation of this tube-like structure, right, from the nucleation center. You have these protofilament-like structures that are formed along the nucleation center, and then they grow by addition of dimers to the ends, forming or elongating this microtubule. The other hypothesis is that it's not just about the cylinder that is growing by adding microtubules, that's growing by adding tubulin subunits. It could be that you have a part of it that is actually growing like a sheet. So imagine now that you have created 13 protofilaments, but like a sheet. It's not actually a cylinder, but a sheet, and you're trying to fold it, much like how children fold a paper. So you could even envisage something like that happening, but that's a little difficult to understand. But there are some very recent papers that are trying to justify this by looking at the kinetics, by doing some modeling analysis to see that this might also be a favorable way of how microtubules are formed. So one is that you have this nucleation center and you're adding protofilaments to it, forming this entire cylinder, and the cylinder grows by adding more dimers to the end. The other is that you have formed instead at the microtubule organization center, at some point you're forming this protofilaments, but you're forming it like a sheet, and at some point it folds. Another hypothesis is that it could be something in between. So you could have the cylinder, but the cylinder is actually growing in terms of a sheet. So if you see this, here is your cylinder, but it's actually growing in terms of a sheet, and then the sheet has to fold. So a number of theories about it, but none of them can either be proved or disproved. But what it shows is that there is a lot of it's a highly dynamic process. You can appreciate that the ends here are very often curled, and we'll come to this curling in a while. So far, do you have any questions? No? Okay, let's go to this. So we have already seen how utilizing a slowly hydrolyzable GTP analog, they were able to kind of show that they can delay catastrophic events during tubular polymerization, right? Now, here is an example where they have used a genetic mutation. And what kind of a mutation is this? So this is a mutation that is in the alpha subunit. Now we know alpha subunit binds to GTP, and somebody had asked me the last time, but so far I still cannot find a paper where GTP that is bound to alpha subunit is hydrolyzed. Maybe I'm missing something. So I would request all of you to actually look up to see if there is anything at all in the literature that actually suggests that GTP in the alpha subunit can be hydrolyzed. So what happens is that you have a mutation here that is associated with the alpha subunit. And because the alpha subunit, so you have an alpha beta subunit. Okay, let me go to the image. It's easier to understand. So you have an alpha beta subunit, right? And when it polymerizes, the alpha subunit is in contact with the beta subunit, such that the GTP cleft is in contact with the alpha subunit. So it has been found that there are some key residues in the alpha subunit that are very essential for the beta subunit to hydrolyze GTP. If you mutate this residue, then this GTP binding cleft in the beta subunit cannot, well, it can bind GTP, but it cannot hydrolyze it. So that's the kind of mutation that we are talking about here, the E254D. So you can appreciate this mutation that is occurring in the alpha subunit and is inhibiting the GTP hydrolysis in the beta subunit. So essentially what it is doing is that it is locking the beta subunit in a GTP bound conformation throughout, which means that you have a very extended GTP cap, right? Now, what they have tried to do in this experiment is to show that if you have such kind of now again it comes back to the same thing. If you do not have a GTP bound lattice structure, then you are inhibiting the chances of catastrophe. And here what they show is you're not just inhibiting the chances of catastrophe, you're also accelerating polymerization. So we'll have a look at the two videos that are here. So in purple is tubulin, and in green are proteins that mark the sort of ends of this tubulin structure. So it gives you an idea कि कितना लंबा है ये tubulin. And this is in solution phase. So what they've done is they've taken something like a glass slide and they've added microtubule or microtubule nucleation sites on the glass slide, right? Otherwise, microtubule, as I said, ये spontaneously polymerize नहीं करता है. Very slow spontaneous polymerization hai. So you have this nucleation sites, and the nucleation sites are arranged in such a way that you will have a bidirectional increase in tubulin structure. So do not think that you also have an increase from the negative end. That's not what it is showing. You have it in such a way that it can grow from both the ends. Now this is how it's moving in the wild type. You see, let's play the video again. So you will see both growth as well as catastrophe events. So I'm not sure if I can, let me see if I can find out one where there is catastrophe. Okay, yeah, I got it. So try to look at here. You would see that this extends and then it kind of, you no longer see this filament. So that's a catastrophe event. So you see growth, you also see catastrophe. You see it forming, and then there is catastrophe is a very quick event, so it just depolymerizes. But if you look at the mutant, the way it behaves now, you will not see catastrophe events, but you will see excessive polymerization, excessive nucleation, and there is not one single catastrophe events, which again hints at the same thing that they had shown previously with a slowly hydrolyzable GTP analog, that if you can inhibit GTP hydrolysis, you are actually stabilizing the lattice structure in the microtubules. You're stabilizing the filament structure, and that GTP hydrolysis, unlike ATP hydrolysis in actin, is not actually promoting polymerization, but it is promoting the defect formations in the lattice, therefore promoting depolymerization. Okay, so come to something else here, but before I go to that, I have a small request to make. So students, I'd like you all to take a pen and a paper, and in the next 10 minutes from now, I'd like you to write down what you understand about actin dynamics and what you understand about tubulin dynamics. This is not a test, doesn't go into your grades, but it goes into, for me to understand whether you have understood what I've said so far. So please take a pen and paper, write down what you understand about actin dynamics, write down what you understand about tubulin dynamics in brief, and take an image of that and send it to me. Hello?
Yes, ma'am.
Thank you. So until then, I'll stop sharing. Okay, students, you have 2 more minutes to complete. You just write in brief what you understand about actin dynamics and microtubule dynamics, and very brief, take an image of that and you can send it to me. Send it to me via email, and if you do not know my email ID, I'm going to repeat it now. It's Anita, ANITA, @iitt.ac.in, or you could send it to me at Anita, ANITA.roy, ROY, @bioschool.iitd.ac.in. And if you are done with it, you can let me know. Okay. हो गया होगा? Okay, students, it's time is up. So stop writing, send whatever you have written so far to me, okay? Hello?
Yes, ma'am.
Okay, all right, let's proceed with the class then. So what we have seen in this video so far is that if you have somehow if you can inhibit GTP hydrolysis, then you sort of lock the conformation or lock the conformation of the individual dimers in such a way in the lattice that you do not create that lattice strain, and therefore there is no way you can actually cause catastrophic events. But so now there is something interesting. Inside cell, you have a lot of GTP. So you can have a lot of these actin alpha beta tubulin dimers, active alpha beta tubulin dimers. But in cells, you do not want growth and catastrophe every time, because you would perhaps want to transport a certain vesicle to the cell surface. And the vesicle is moving along rail tracks or moving along these microtubules to the surface. अगर बीच में ही चलते चलते माइक्रोट्यूब्यूल कैटास्ट्रोफ हो गया तो ये वेसिकल पहुंचेगा कैसे सेल सरफेस तक, या फिर यू आर ट्राइंग टू separate the chromatids, right? And you have an entire microtubule that is helping to move this chromatid undergo complete catastrophe. It's just falls apart. If that happens, then how are you going to pull the chromatids apart? So cells में ऐसे नहीं हो सकता कि you have tubulin molecules which are undergoing such catastrophes every time. So things inside the cell have to be regulated, much like the way actin polymerization was regulated inside the cell. So I'm going to give you a couple of examples of how inside the cell the microtubule dynamics is actually controlled. And one of the beautiful example actually lies here in this video. So these green structures that are seen to light up the end of these microtubule filaments are actually proteins called end-binding proteins, EB. And these end-binding proteins, as the name suggests, binds to the end of a microtubule, a polymerizing microtubule. And these have been tagged with GFP, so you can see them glowing green. And you have a lot of these proteins that can sort of modulate the dynamics of a growing microtubule. So you have some proteins which can assist in polymerization. Okay, so polymerization proceeds at a certain rate, but inside cell you would find that microtubule polymerization is actually much faster. The reason it is much faster is because you have some proteins which are called microtubule polymerases. As the name suggests, it helps in polymerization of the microtubules. So these bind to the plus end of the microtubule, growing microtubule end, and they bring in these dimers and help in quick polymerization of the microtubule. So they have an affinity for the plus end, and they bring the dimers and they help in quick polymerization. So these group of proteins, these microtubule polymerases, bind at the tip, right? And then they leave. You have other proteins called these end binding proteins that seem to behave differently in different conditions. So I'm not going to go into great details about it. But what is seen is that rather than binding absolutely at the tip, they'll bind a little behind. So how is it, or how do we look at these end binding proteins that were actually shown here in green? What is now thought is that you have this end of a microtubule where you have polymerization events occurring, and then you have a certain region where the GTP has been hydrolyzed, but again, like actin, it's, you just take the analogy of actin. The GTP has been hydrolyzed, but the inorganic phosphate has not left the cleft. So then it gives rise to a different conformation which is somewhat stable, more stable than this GDP bound form. It's the same like actin, right? Because this GDP bound lattice is actually under strain and would like to depolymerize. But this, on the other hand, when it is bound, so this part, which is GDP plus PI part, is what binds to the EB1. So it definitely binds to the end, but not at the end where polymerization is occurring. Not where the new subunits are being formed, but a little away from that. So here is again another example. You have this microtubule polymerase binding absolutely at the tip where it is bringing the new dimers. And then behind that, right behind that, you have EB1 binding, which helps in sort of, it does promote polymerization in certain cases. But then if these themselves are post-translationally modified, then they can also promote catastrophe events. So there are multiple layers of control. Don't want to go into detail, but just giving you a hint at this. So you have a way to increase the size of this GTP cap, because if you increase the size of the GTP cap, you are avoiding catastrophic events. You're also inducing a very fast microtubule polymerization. You have this EB1 binding site which somewhat promotes polymerization, but if you post-translationally modify EB1, then it can also promote catastrophe. So you have another layer of control, right? Okay, so another layer of control is by some motor proteins, and we will, for today's class, we'll call them kinesin. We'll come into the details of these motors later on. So for today's class, let's think of them as some proteins that are sort of walking on microtubules. So there are some proteins which are walking on microtubules, some classes of kinesins, but there are other classes of kinesins which do not really walk on microtubules, but which like to bind to the plus end or the growing end of microtubules. So both of them can actually affect the way microtubules are going to polymerize and depolymerize. So here is a family of kinesin called kinesin 13. And this has been found to be a kinesin that does not quite walk on microtubule. Rather, it likes to see the plus end of a microtubule. And what it does is it uses, so here is a structure of what it actually does. Now, before we go into this, let's look at this. So this is an electron micrograph image of a microtubule, right? And you see the ends have curled up. And we understand that when catastrophe occurs, it occurs by curling of these protofilaments. So the straight protofilaments are the ones that would be present in a growing lattice. But if you have these kind of curling events, then it is promoting catastrophe. So then the simple idea is that, okay, if I can promote the curling, promote the buckling event of a microtubule protofilament, then I can promote its depolymerization. Very simple. You have catastrophe occurring where the protofilaments buckle like this, and they buckle and then they dissociate, and it's very quick. It's not one single subunit being removed at a time, but really the entire thing just collapses because there is buckling. If that is true, then in cells, if you can somehow promote buckling, you would promote catastrophe. So that is exactly what is being done by these set of kinesins. And this is, so if you now take, so it's again the same principle. You take microtubules and you take these kinesins, which like to be at the plus end. That is at the end where you have these structures which are bound to GDP or the GDP plus PI. And you look and you take a lot of these images, cryo-electron images, and then you try to combine all of these images to see exactly how it forms, and you have this kind of an average image that you see. And what do you see? You see that these individual kinesins are actually promoting buckling of the microtubule protofilament. So microtubule protofilament seems to buckle at the kinesins, which indicates that these kinesins are actually promoting depolymerization, but by inducing that kind of a buckled, that kind of bending, called a ram horn. Ram horn because the horns of a ram, the sheep, are actually bent like this. So it kind of promotes that conformation. So you have one instance of a motor protein that is utilizing energy of ATP hydrolysis to actually bend and buckle microtubule. You have other family of kinesins like this, which actually seem to destabilize the entire lattice structure because of their walk itself. So what happens when they are walking on microtubule? There is actually some interactions occurring between the amino acid side chains of this motor protein kinesin with the amino acid side chains that are present on the microtubule, right? So then there is a conformations change every time it steps, and the steps are released. So there is certain things that is happening on the microtubule too, and that can also destabilize the microtubule. But the best example that I found for all of you, the best visual example was that of kinesin 13. So please remember this, that if you can increase buckling, if you can increase the bending, you can increase depolymerization of a microfilament. Okay. So with that, we come to another concept. Now, this is again a concept. So, but there are some indications to it. So in cells, you will have an actin being formed. You will have microfilament. You'll have actin microfilaments. You'll have microtubules being formed. But it has been observed that very often these microtubules seem to grow very close to these actin filaments. It's as if the actin filaments are providing a guide rail on which the microfilaments are growing. And to understand this, people were trying to see what could be the thing, and they found out a protein called the spectralacin. It's not important to know the name of the protein, but rather important to know that there is a phenomenon of this kind. So you have something here that binds to these end-binding proteins on microtubule and also binds to actin bundles. And therefore, it provides a way by which a growing microtubule might follow an actin bundle. So it's kind of telling the microfilaments that, okay, you need to follow this path when you're growing. So it's a directed motion of growth of a microtubule. So growth of a microtubule, as I explained initially in the class, growth of a microtubule can also exert force on a bead, just like actin polymerization. So So similar studies, just like actin polymerization, have also been done in microtubule polymerization, where they have taken a bead and you attach microtubule bundles to the end of the bead. And then you have a rigid wall. And again, you see that because of end fluctuations, you can attach more tubulin dimers to the end, and that would start pushing the bead. And therefore, they could calculate the amount of force generated by the microtubules. Now, if you have a single microtubule here, the force generated is low. But if you have bundled the microtubules, if you take a lot of them, bundle it together, then you have a huge force that can be generated, just like in actin. If you have one single filament, the force generated is low, but if you have a number of them, then the force of polymerization is it's cooperative in nature, and it just spikes up. Okay, with that, yeah. So this is something that I missed while speaking about actin, but I'm going to just give a little briefing here when I'm talking about microtubules. So there are some, yes.
10 minutes are remaining.
Okay, thank you so much. So I'm almost at the end. Thank you. So, so there are some organic molecules which are known to bind to tubulins, and which can either promote its polymerized conformation or promote depolymerization. So, for example, agents like Taxol here, they bind to sites between two adjacent. So there are protofilaments. So these are the protofilaments. So sites in contact between two protofilaments, they bind there, and they seem to stabilize the structure. So if you want very stable microtubules in solution, you add a little bit of Taxol. But if you want to destabilize the structure, then you can add some other agents such as colchicine. And colchicine is known to bind to in a polymerized microtubule in a site that is between the alpha and beta subunits. So between these sites, there is a region where colchicine binds, and it sort of helps in what should I say? It sort of helps to kind of induce a very strained conformation and therefore buckling of the protofilaments and dissociation. So you have a lot of molecules that are used to actually understand how microtubules function. In fact, if you remember the first slide of microtubules where I was talking about Shinya Inoue and his pioneering work with microtubules, he had actually used colchicine to study how microfilaments form in a dividing oocyte. So how they form when that oocyte actually divides and undergoes mitosis. Now I'm going to stop the class here, but I'm going to give you something to think about. So, and maybe we can discuss this in the next class, or maybe you can just carry on and discuss it here. So you have a situation, so what we have known so far is that microtubules can polymerize, depolymerize as catastrophe. There are other proteins that can sort of inhibit, quicken polymerization, can enhance or can induce its depolymerization inside a cell. Now you have a situation where a cell is dividing, right? And it has formed these X-like structures, metaphase chromosomes, and they're perfectly aligned at the metaphase plane. And then you have these spindles that have originated from the two poles, from the microtubule organizing centers, and they have come up and they have attached to the kinetochores, or the point where these X's are linked to the microtubule. And now, at the right time, these two chromatids need to be pulled apart to the pole, right? Now, we can kind of understand that, okay, because the microtubule organizing center is known to produce microtubules, it might produce a lot of these microtubules, and one of them might capture a kinetochore and bind to it, and that binding would be quite stable, and therefore it's attached to microtubule. But then what is it? What is causing these what is causing these chromatids to be pulled apart? We all know they're being pulled apart, right? We've seen videos where the chromatids are being pulled apart. But how do you create that force of pulling it apart? So far, we've talked about microtubule polymerization, the force it exerts. That's also force. But here, what we are talking about is that these chromatids need to be pulled apart. What do you think, or how do you think this is being pulled apart? There's a lot of force that is required to do this. How do you think this might be happening? Any clue, any idea? Just anything that comes to your mind?
Ma'am, I believe. Hello.
Yes, yes.
So, ma'am, what will happen basically at kinetochore? Maybe the plus end will bind to chromosomes by their plus end, right? So during the after the post metaphasic event, like pre-anaphase, what will happen? Microtubule will start catastrophe, so that they will shorten and will pull the chromosome from center to poles.
Okay, but then if there is catastrophe, you understand catastrophe is like a complete, so
Yes, ma'am.
microtubule catastrophe and microtubule catastrophe is, it is exactly as the word goes. It is catastrophic. That means you have depolymerization. You have a complete breakdown of that structure. So do
Yes,
you think
ma'am.
there would be catastrophe? Because it has to be stable, it has to pull it. If it just पूरा अगर खुल ही जाए पूरा structure, तो वो pull कैसे करे? Part
Okay.
of it is right, but not catastrophe. It's not catastrophe.
Okay.
How do you think it might happen then? A plus end तो bind करके बैठा है ना?
Then, ma'am, there might be certain other factors that will like remove these monomers from the plus end at a certain rate that is required for the pulling apart of these chromosomes.
Okay, okay, okay. Thanks,
Ma'am,
Farman.
bending this
Yes.
microtubules,
हाँ क्या?
bending of microtubules can generate force.
Yes, yes. I would take both of your answers and thank you so much. So you're both right in a way that first of all it's not going to be a catastrophic event because if there is catastrophe तो पूरा collapse कर गया वो नहीं हो सकता. But it indeed is something to do with the microtubule itself. There is no other. There are motor proteins there, but even if you remove those motor proteins, it still works. So it is primarily driven by microtubule depolymerization, slow controlled depolymerization, not catastrophe. And what happens is this bending, ऐसे जो bend हो रहा है, that seems to be having some force on the structure that it is holding. So think of it like a puppet. I mean, even if you have not seen a puppet show in front of you, but you might have seen it on television. So think of it like a puppet. Puppet का क्या होता है? You have strings attached, right? At the hand, at the neck, at every position, and you pull one string and you see it moving. So if you have a string that is held somewhere here, you can easily move the head, right? So it's something like that. When the microtubules are buckling, that should also give it some space for these filaments to sort of polymerize slowly, because there is this buckling event and the buckles keep forming. It's going to be very, so you have to imagine things to be very dynamic first of all. ऐसा नहीं है कि जो प्रोटोफिलामेंट बकल कर गया वो ऐसे ही बैठा रहेगा, उससे भी डिपॉलिमराइजेशन एंड से हो रहा है, and this buckle thing is forming, and you have this receding, right? At the same time, you will have some proteins which are there on the kinetochore, some fibrillar structures, because even that has to have some amount of elasticity to it, because this cannot be a rigid structure that is binding. There's dynamism here. So if that binds to the end to these buckled structures, and this is actually going down because there is depolymerization occurring and the buckled structures are going down. So if in effect what it does, it drags this entire chromatid together. And there have been, so as I said, this is not something that can be easily envisaged, not something that can be easily sort of understood in that sense. But there are some very beautiful experiments that have been done to see how much of load can this microtubule carry. So what turns out that the load it can carry is quite enormous. A single microtubule can carry an enormous amount of load, almost like an ant, which can carry like some multiple times of its own body weight. And then this buckling, both the buckling and the depolymerization, not catastrophe, but buckling and depolymerization together is creating enough force, because there is a tethering between the chromatids and this microtubule ends, and you have the microtubule ends constantly depolymerizing, and this whole thing sort of moves slowly down and pulls the chromatid along. So maybe for the next time, I will share a video of this experiment that was done, so that it becomes a little clearer. It's as I said, microtubule dynamics is still something that is being hotly debated. It's not very well understood, especially how it is going to be formed, whether it's a sheet-like structure that collapses, whether it's a tube that is getting formed, or whether it's a mix of both. It's highly debated. It's still not very well known. So yeah, so this is all about microtubules and microtubule polymerization for today. Any questions you have, I can answer them now. Please do not forget to send me whatever you've written, so that I have that in the next 15 minutes. Okay, I'll stop presenting and
I have
I'll
a
take
question
any
actually.
questions you have. Yes, if you have any question, go ahead.
Ma'am, I have a question.
Yes.
So, ma'am, as you said, like it will not be catastrophe, it will be depolymerization, right? So,
Yes.
how does this depolymerization is controlled? Like, are there certain factors that are controlling it? Or, like, is it intrinsic to microtubules? Is this property intrinsic to microtubules that they can control their depolymerization when
You're asking
pressured?
me how is it going to be a controlled depolymerization and not a catastrophe event?
Yes, ma'am.
Okay, so
Yes, ma'am.
honestly, we do not have complete answers to this. What we know is that there are molecules that could be acting at the ends, and that could be removing individual subunits, right? Now, this is again something that we kind of have a little pictures from here and there. We do not really know exactly what is happening. You have to appreciate that the kinetochores themselves are huge complexes of more than 100 proteins. You have a lot of motor proteins acting there. There are ideas that you can have fibrillar structures that are connecting the kinetochore with the microtubule ends. So so the motor proteins there could be doing this and could be doing it exactly at these ends, and also there could be proteins that are actually inhibiting catastrophe formation, but still you would have that small buckles happening. So we do not quite know the entire picture. Honestly, we do not. This is still an area of research. We do not know the entire picture. But we have some bits of ideas that this is perhaps what is happening, because if you have a catastrophe event, you would have a complete collapse. You do not have that. You need to have it absolutely, how do I say, it has to be controlled. If all the protofilaments start to fall apart, then you cannot have this being pulled. So catastrophe nahi ho sakta, but isko shorten bhi hona padega. So we can envisage that there should be something that is actually taking out these subunits, helping in the buckle formation, but the buckling is not to the extent that you will have a catastrophic event. So we can envisage this. We do know certain things that are there in the kinetochore, say some motor proteins that are there in the kinetochore that are actually walking on the microtubules at certain times. It's not always maybe at the start when the separation has to take place, there are some motor proteins that start to walk away from the kinetochores. Things like that are known, but the total picture is not very clear. I honestly do not have a complete answer to this.
Okay, ma'am. Thank you.
Okay. All right, students, please do send me by email whatever you've written. That will help me understand if you have understood. Okay, and then we will move on to motor proteins in the next class. All right.
Yes, ma'am.
Okay, okay. Thank you so much. Thank you. I hope you've noted my email ID so that you can send me the whatever you've written by email. Thank you and see you. So today is Friday, right? See you on Tuesday.
Yes.
Thank you.
Thank you. Bye.
Thank you.
Thank you, ma'am.